Good evening. My senior research project is on microfiber cloth's efficiency on microbial removal. A little bit about me. My name is Giada Pugliese and I attend Tunstall High School. I plan on attending JMU next year and majoring in psychology while minoring in pre-occupational therapy. Why this topic? Foodborne illness rates are continuing to rise all along the U.S. The Norix brand is also growing in popularity due to their use of non-chemical cleaning agents. The Norix brand is specialized due to their use of silver antibacterial particles and microfibers, which allow them to lift and kill the bacteria as soon as it is off of the surface. This topic has a personal connection to me because my father owned small restaurants all along the city. This allows me to understand the cleanliness of inside of a kitchen. My mother is also an advocate for the Norix cloth brand due to their use of non-chemical cleaning agents. Foodborne illness is a growing problem all along the U.S. The symptoms and strands have worsened, causing more sickness and illness all along. The common symptoms of foodborne illness are vomiting, nausea, fatigue, and joint backaches. Foodborne illness can be prevented if preparation areas are taken to regularly. A proper food safety culture allows a customer to feel comfortable while eating in an establishment. In order to obtain a proper food safety culture, the establishment needs to wipe the surfaces regularly and store all food in the right areas. Cross-contamination is caused when a food item is being prepared on a surface and it is not wiped down before the next food item is being prepared. The most important factor in microbial removal is the durability and effectiveness of your cloths. The cloth is what you use to wipe the surface and what is used to kill any bacteria on your surface. The durability of these cloths depend on how many times they can be laundered. A microfiber cloth by the Norix brand can be laundered more times due to their use of non-chemical cleaning agents. Bacterial adhesion is how strong of a force a bacteria has on a surface. A biofilm can form on top of a bacteria if it is left not washed off. This will cause an automatic increase in cross-contamination because without the use of harsh chemicals, it is difficult to remove. My study is filling research on how Norix cloths compare against generic cotton cloths when regarding microbial removal. This will be helpful when discovering which cloth you should use when cleaning workplace surfaces. My research question is, do microfiber cloths kill more bacteria than generic cotton cloths offer workplace surfaces? My null hypothesis is if Norix cloths are used to clean a surface, then they will show no statistical difference than generic cloths. My alternative hypothesis is if Norix cloths are used to clean a surface, then they will show lower bacteria counts than generic cotton cloths. I could come to this conclusion by previously researching the effectiveness of Norix cloths compared against microfiber cloths. The variables in my experiment that were controlled were my surfaces, and that they were kept constant were my temperature. My dependent variable was my bacteria type, and my independent was my cloth type. The first step in my methodology was to gather my materials. The materials needed for my experiment were Escherichia coli, E. coli, Sarcina aurantiaca, S. aurantiaca, generic cotton cloths, Norix cloths, and 2 stainless steel tables. The sample size of my experiment is 88, and my sample population is 44 squares of E. coli and 44 squares of S. aurantiaca. Before beginning my experiment, I divided 5 Norix cloths and 3 generic cotton cloths into 44 squares. After, in order to ensure cleanliness of my surfaces, I bleached, rinsed with boiling water, and then went in with a hypochlorite sanitizer. After air dried, I taped 44 squares along the surface and labeled them 1 through 44. After, I placed a drop of distilled water into each square in order to allow my bacteria to spread more evenly. After, I took a sterile cotton swab, dipped it into my bacteria, and placed it all along my surface. In order for me to ensure randomness, I assigned each square type a different cloth number. This allowed me to make sure that my experiment did not contain any kind of bias. After, I dipped each cloth type square into water and cleaned the surface. I cleaned the surface using the sweeping method, which is going corner to corner until making a full square and then filling in the center. After, I used a sterile cotton swab, swabbed the surface, and placed it into the correct Petri dishes. All of my E. coli samples went into nutrient agar, and all of my S. aurantiaca samples went into lauryl triticose agar. I placed both sets of Petri dishes into the incubator at 37 degrees Celsius for 48 hours. My experiment did not show a zone of inhibition due to insufficient growth of bacteria colonies. I also failed to reject my null hypothesis due to the failing to gain a comparative on generic cotton cloths and Norix cloths. At the 0.05 level of significance, there is not sufficient evidence to conclude that Norix cloths are significantly different than generic cloths. The flaw causing the lack of data is the use of bleach as a sanitizer prior to my experiment. The use of bleach as a disinfectant is very common. Bleach is up to 99% effective, which is why there were no growth on my Petri dishes. Bleach also lasts for up to 24 hours. My surfaces were not allowed up to 24 hours before my bacteria was placed. But bleach also kills bacteria within 30 seconds. My experiment could not take place in under 30 seconds in order for my bacteria to remain alive. The results of my experiment differ from our prior research because my surfaces needed a prior sanitation. In previous research, their surfaces were kept in controlled environments or they were either recently purchased. My surfaces were borrowed and kept in previous uncontrolled environments. The largest limitation in my study is the lack of research around bleach as a surface disinfectant. Another limitation is how much time that I allotted in order for my experiment to take place. When conducting further research, I recommend to lower the potency of the bleach by diluting it with water. This will allow the activity to lessen, causing the experiment to be able to take place in my given time frame. The next thing that I would recommend is to allot more time in order for this experiment to take place. This experiment taught me how to work on my time management skills. It allowed me to create schedules and manage little bits at a time. It also taught me how to work on my organizational skills. It helped me through the inquiry process by being able to manage my thoughts and different methods and tips such as brainstorming. I would like to thank a couple people for helping me with my SRP. Firstly, I would like to thank Ms. Long for helping me learn and go through the inquiry process for the past 2 years. I would also like to thank Mrs. Gravitt and Mrs. Dillon for helping me through the statistical and science aspect of my experiment. Lastly, I would like to thank my dad for allowing me to use his workplace surfaces in order for this experiment to take place. Does anyone have any questions? Yes.
I think I heard you say that you did not discover any research regarding the use of bleach on the surfaces, so why do you think that is?
Mrs. Walsh asked why they never used bleach previously on the surfaces.
Is that what the research you said you discovered, was that?
Can you rephrase your question? I'm
Yes, so
sorry.
I thought you said that you had discovered that there was no research about using bleach on the surfaces prior to this kind of experiment. Is that what you found?
Ms. Waltz asked if I found no prior research on the use of bleach. Before doing my experiment, I didn't research the use of bleach. I didn't think about it. It wasn't within the limited scope of mine. It was more of a broader aspect in my experiment.
So you said on your slides that your mother uses Norix.
Yes.
So she must be a believer that it has some significant or some type of influence on cleanliness. So given that, and then bleach, do you believe that there's a difference, or do you kind of stand behind your study that both the cotton versus Norix had the same effect on cleanliness?
Dr. Jones asked if I still believe that the Norix cloth would be statistically different if this project was done correctly. I believe that the Norix cloth does show lower bacteria counts due to their use of the silver antibacterial particle, because that's an uncommon thing in the cloth, and that's what sets it apart from the other cloths.
Okay. So if I'm cleaning, why shouldn't I just use a cotton cloth and bleach to clean?
Because Dr. Jones asked why I do not just use bleach instead of a microfiber cloth. Bleach is a very harsh chemical, and it cannot be used continuously because if a food item is prepared on top of bleach, it is not safe for human consumption.